Hello students, now we discuss the next chapter, that is chapter 9, which have heredity. Earlier this chapter was heredity and evolution. So evolution is related, and in this session your physics of 25 marks, chemistry of 25 marks and biology of 30 marks. So here world of living having 23 marks this unit, and natural resources having 7 marks. So altogether 30 marks question will come from the biology sections, and 25 that is the physics and chemistry, means that is 80 marks. So in this chapter we will discuss about the heredity, and in this one, that is what is variation, what is character, what is trait, and after that we will discuss about the different type of types of activities, Mendel's law, chromosomes, and sex determination, etc. So we start this chapter from this one. So say heredity means what? What is heredity? So how you define it? Heredity means it is the transmission of genetically based characteristics from parent to offspring. Means from parent what we got, that is the heredity. So heredity may be either disease can be hereditary, okay, they can transfer from one to another, and some positive characters, negative characters also come from our parents to us. So this is also called heredity. So say transmitted means genetically transmission of characters is heredity. Now see variation means what? Variation is the change. It means very small, here given small differences among individuals are called variation. So see small difference. So suppose anyone having 5 fingers, and one of the additional finger come to other person's hand, that is called variation. So slight variation takes place in organism. So small differences among individuals are called the variation. This is the variation which is very minute, but it is occur in nature due to some changes. Here character and trait. Okay, character means any inheritable feature is a character. Something which are inherited from one to another generation, one generation to another generation is called character, but trait is the alternative form of the character. Alternative form means we can say that character, if character of eye color, it may be trait, that is either it is black or blue, contrasting, that is contrasting character, either black or blue. Here hair shape, it may be curly or straight. Curly hair or straight hair is the hair shape, that is hair shape is a character, and curly or straight is a trait, we can say contrasting character. Ear lobe, sometimes ear lobes become free, that is free, and sometimes attach. So free or attach ear lobe, that is also the trait, free or attach, and character is ear lobe. Eyebrows, some persons having the very dense eyebrow like Krur Singh, and some person having thin eyebrow looking very good. So this is the eyebrow, they are contrasting traits, that is the bushy, heavy bushy, or that is the thin. Now we discuss next thing, that is the chromosomes. See, you know chromosome, where it is found? Chromosome is the is the structure present inside the nucleus of the cell. So inside the nucleus there is chromosome, and this chromosome is having the genetic information carrier, or they carry characters from generation to generation. So chromosome is the hereditary unit of us. In this one DNA is present. So chromosome, the carrier of heredity. Chromosome, it is the carrier of heredity, carrier carrier of heredity. Of heredity. This is the chromosome. So chromosomes are highly condensed carried chromatin fiber, coiled chromatin fiber. Inside the chromosome means if you want to go towards the chromosome, you have to reach. So here this is the cell, and inside the cell there is nucleus, and these are the chromosomes inside the cell. So you have to go towards the chromosome by moving towards the nucleus. Inside the nucleus there is a chromosome. See, its number is fixed in different organisms. Their number is in human beings it's 46. So when we can say diploid means 2n, it is of 46, or we can say 23 pair. And haploid, other one is haploid. Haploid is n, it is of 23 only. Okay, so this is the half number of chromosomes, this is the full number of chromosomes. So diploid is present in every cell except the gamete. So haploid and diploid are the characters which is found in the body of human beings. Either they are gamete or they are the general cells. So when there is gamete, there are gametic form, then we understood that they are half number of chromosomes there. So say out of 23, this 23 pair, so autosomes are 22 pair. Autosomes, and allosomes are 1 pair. Okay, so these allosomes are also called sex chromosome. So they are participating in different activities of sex determination, we can say. So here in male, these allosomes are XY, and in female these are XX. So say male having the XY allosomes and female having XX allosomes. So in sex determination we will use this one for male or female child. In human beings, there are 46 chromosomes, you understood, and XX pair similar parents is found. Now, how sex determination takes place? So, see, in this one, there is the male, there is the female. So, this 22 will not useful for this activity. Here, 22 is not, but this last pair, one pair which have XX chromosome is required. This last pair, that is XY chromosome, is required for which purpose? For the sex determination. So if this Y and this X, so XY comes and fuse, then they form the male. And when this X and X will fuse, they form the female. So this is the way of sex determination in the human beings. So sex determination by the help of this activity. So determination of sex mainly depend on the last pair of chromosome, that is the allosomes, and if the male's Y will active, fuse, they form the male child. If the X of the male is active, they form the female child. So this is the sex determination in human beings. It takes place in the natural way, means no one can No one can have the capacity to do the activity of Y and X, that is means they are unable to form that one, means male child or female child, in an artificial way. So it is properly natural way, and we should not claim, means in our society that claim for the female child to the to the female parent. So this is really not a scientific activity, means we cannot blame the female for the female baby birth. So this is the male and female, that is determined fully by the help of nature. Now we understood about some technical terms related in this chapter, that is gene. The first one, gene. You know gene? Chromosomes unit. Okay, so chromosome is on-self unit of hereditary character, and they are made up of some units. They are called genes. Genes are the specific part, DNA segments of chromosome. The first, here you say G-E-N-E, gene. It is the specific part. It is the specific part of a chromosome, chromosome, that is called gene, which determine the hereditary character. Gene determine which determine the hereditary character. Hereditary character. Okay, so see, this is the gene, and there are nearly 30,000 genes in humans. In our body, 30,000 genes present in human body. So nearly approximately 30,000 genes present. So it is the DNA segment, and genome is the full component of a DNA. So Genome is genome. It is it is the full complement of DNA. It is the full complement of DNA. Of DNA. Okay, and in which includes all the genes and intergenic region of an organism. Of DNA includes all genes and the intergenic region of organism. Intergenic region of organism. Region of an organism. Understood? So genome is the full complement of DNA. So this DNA includes all genes and intergenic region of an organism. After that, this is the alleles. Understood or heard the name of alleles? Allele alternative forms of a gene called allele. Alleles. Alternative forms of a gene occupying alternative forms Alternative forms of a gene occupying the same occupying the same uh, position on homologous chromosome, on same position on homologous chromosome, homologous chromosome and affecting the same characteristics but different ways, and affecting the same characteristics in different ways. That is a pair of chromosome showing four imaginary genes, that is ABCD. The situation depicted here are AA homologous, BB homologous, CC homologous. See here, homologous means what? Homologous means same, like ways. Here you see, when we say round seed, this is homologous. When we say this is the round seed character, round seed, this is the homologous pair. When we say RR, wrinkled seed, WRINK, wrinkled seed of P, that is homologous. Okay, both are homologous. Homo means same, homologous. But when we say capital R, small r, it is dominant as round seed. Round seed. This is the heterologous, which is heterologous. Understood? So homologous means the same and heterologous means the different. So two things more, that is genotype and phenotype. Means when we study the gene, that is the characteristics based on gene is called genotype. Characteristics based on the outer character, means physical or features, is called phenotype. See, genotype. Characteristics based on, see, the set of gene present in the cell of an organism. Set of gene present in a cell of an organism, is genotype. Okay, in this one the genetic constitution are called genotype, that is RR, this is, and this is the genetic constitution of non-neural. Means say example, RR, these are the genotype. Okay, and phenotype is different from it, that is outer character. Phenotype, it means this is the external character. Phenotype is the observable characters which is genetically controlled. Observable characters which observable characters which is genetically controlled, which are genetically controlled, that is phenotype. Phenotype means we can see from outside. See, the expressed shown, it is the expressed character, shown character of the offspring on the monohybrid cross of the genotype ratio, that is 1 to 1, and the phenotypic ratio, that is 3 is to 1. One example here can clear this genotype phenotype. When the monohybrid cross takes place. So here you see round seed, wrinkled seed, wrinkled seed. Okay, what happens? Here RR, here RR. So this R will come, this R will come as a gamete, and when they fuse in F1, that is the R. So here you see this is phenotypically round. Okay, so phenotypically round seed comes here. So this is in F1 generation. Okay, and genotypically they are having both the character. Round seed character is here R capital, and wrinkled seed character is R small. So you can say what we observe from outside. So they are outside round seed having character. So we can say it is phenotypic character. Characters outside, that is phenotypic character of gene, means we can say what set of gene present in a cell of an organism, that is the genotypic character. Understood? Now Mendel's experiment on inheritance. See, Mendel do experiments on the different type of pea plant, and they provide us monohybrid and dihybrid cross, and on the basis of these two crosses they give 3 of their postulates, means their principle, or we can say law. So inheritance, Mendel provide, because Mendel is also called the father of genetics. So genetic laws they provided to us. So Mendel, the basic principle of genetics were discovered for the first time by the Gregor George Mendel. So they provide us the idea about how the things become interact to each other, and after crossing with the pure parent they have the changes, and these changes either shown from outside, that is the phenotypically changed, and it is shown from the inside, but after some times they also segregate. So 3 laws given by them in this inheritance, law of inheritance. First one, law of dominance, second one, law of segregation, and third one, law of independent assortment. That is 3 law will be, we will discuss these 3 laws one by one. So see, Mendel, what they do, the monohybrid and dihybrid cross. First we understood what is monohybrid and what is dihybrid cross. Monohybrid cross means the cross between the 2 parent having the single characters, they taken the single characters. Monohybrid cross. Okay, so the cross between the 2, the cross between the 2 pure individual, pure parent having single character, single contrasting character. Okay, so in this one you will see, so example, round versus wrinkled seed of pea, white flower versus violet flower. Next one, that is either the green or yellow seeds color. green versus yellow seed color. Yellow seed color of P. Understood? So these are the monohybrid cross. But when we discuss about the dihybrid cross, there are pair of characters. So in dihybrid, you can see more than one character is there. See, dihybrid cross. The cross between two pair of characters. Two pair of character. Example, round and green or round yellow versus wrinkled green. Wrinkled green. This is the two pair of contrasting characters they can do the experiment. So in monohybrid cross and dihybrid cross, in both cross they can take the characteristics of different type of pea plants and then they do the experimentation. After experimentation they have the genotypic and phenotypic ratios. So in later class or next class we will discuss about the monohybrid dihybrid cross, and we also make the Punnett square checkerboard and understood about the things. After that we will discuss all the three laws of inheritance. Okay, thank you.